I welcome the researchers, scholars, students, and scientists all over the world. This is Advanced Multiple YouTube channel. My name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development in Corporation. Okay, well, today I'm going to share my knowledge, my experience, and my research under the title of History and Introduction of Monoclonal Antibodies. For this purpose, I collected the scientific information and the data from open source like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine US, MBAS, directly of Open Access Journal, ProQuest, CNKI, that stands for the China National Knowledge Infrastructure. Some of the information collected from the ResearchGate and Crossref. Before moving forward, I would like to talk a little bit about antibodies. An antibody or immunoglobulin is Y-shaped protein produced by beta cells or B cells that is used by immune system to identify and neutralize foreign objects like bacteria, viruses, toxins, or foreign bodies or foreign objects. An antibody produced by single clone cell called as monoclonal antibodies. Monoclonal antibody is therefore a single pure type of antibody. Antibodies are secreted by a type of the white blood cells called as plasma cells. Each antibody recognizes a specific antigen unique to its target. Monoclonal antibodies are identical because they were produced by one type of the immune cell or single parent cell. So polyclonal antibodies are antibodies that are derived from different cell line, and different in amino acid sequence. All current therapeutic antibodies are of immunoglobulin class when the objective of antibody therapy is to directly kill the target cell. Okay, well, history of monoclonal antibody development. In 1964, Littlefield Ward developed a way to isolate hybrid cell from two parent cell line using the hypoxanthine aminoterine thymidine selected media. In 1974, Kohler and Milestone provided the most outstanding proof of the colonal selection theory by the fusion of normal and malignant cells. The Nobel Prize in Physiology or Medicine in 1994 was awarded jointly to Neil K. Journey, George J. F. Kohler, and Cesar Milestone for theories concerning the specificity in development and control of the immune system and discovery of the principle for the production of the monoclonal antibodies. Okay, well, these are the three scientists who play outstanding, amazing role to develop and produce the monoclonal antibodies: Cesar Milestone, George J. F. Fuller, and Littlefield J. Ward. Okay, well, there are the three main type of the monoclonal antibodies designed nowadays all around the world. The first one is the murine source monoclonal antibodies, then chimeric and monoclonal antibodies, and third one is the humanized monoclonal antibodies. The first one actually is rodent monoclonal antibodies with excellent affinities and specificities generated using conventional hybridoma technology. Clinical efficacy compromised by the MAMA human anti-murine antibody response, which lead to allergic or immune complex hypersensitivity. Chimeric monoclonal antibodies actually are the chimers combine the human constant region with the intact rodent variable region. Affinity and specificity unchanged also cause human anti-chimeric antibody response approximately 30% murine source. Okay, well, the third one is contain only the complementary determined region CDRs of the rodent variable region grafted on human variable region framework. Okay, well, what is the advantage of monoclonal antibodies? Monoclonal antibodies having five major advantage. The first one is the homogeneity. Monoclonal antibodies represent a single antibody molecule that bind to antigen with the same affinity and promote the same effector function. The second advantage is the specificity. The, the product of the single hybridoma react with the same epitope on antigen. The third one is immunizing antigen need not to cure the characterized and is ultimately not needed to produce large quantity of the antibody. Fourth one is the selection. It is possible to select the specific epitope specificities and generate antibodies against a wide range of the antigen determinants. The fifth advantage is antibody production. Unlimited quantity of a single well-defined monospecific region can be produced on commercial or business purpose in for the utilization in clinic and hospital. There are also some disadvantages associated with monoclonal antibodies. There are the five disadvantages. First one is affinity. Average affinity of monoclonal antibodies are generally lower than polyclonal antibodies. Effector functions. Because antibody is monoclonal, it may not produce the desired biological response. The third one is the specificity. Monoclonal antibodies work against conformational epitopes or native proteins, may lose reactivity with antigen. Fourth disadvantage is cross reaction. Antibodies sometimes display unexpected cross reaction with unrelated antigens. Fifth one, fifth disadvantage is the time and effort commitment. There are a lot of efforts and commitment needed to produce the monoclonal antibodies. Okay, well, this is the regular normal antibody. Antibody structure actually comprised of the two main chain. The first one is light chain and heavy chain. Light and heavy chain, they are connected with each other by disulfide bond, and it have two segments or the two part. The first one is variable, second one is constant. Constant segment having the constant molecular structure that's not be changed. The variable segment actually change the chemistry, change the structure all the time because it have to bind with the antigen. So because of its variability or changing the molecular chemistry or structure, it can bind and destroy the different kind of the antigen. Okay, well, this diagram actually highlights the types of the monoclonal antibodies used in different industry, or you can say that there are the seven different kind of the monoclonal antibodies available to use for the clinical or research purpose. The first one is the glycomodified monoclonal antibodies. The second one is altered amino acid in constant region. The third one is use different human monoclonal antibody isotope. The fourth one is link isotope to monoclonal antibody with stable linker. The fifth type of monoclonal antibody is link drug to monoclonal antibodies with cleavable linker. Sixth one is insert DNA to monoclonal antibodies variable region, fuse to signaling peptide into the T cell to induce expression of CAR. CAR stands for the chimeric antigen receptor. The seventh one is cross-link region from two monoclonal antibodies. Okay, well, in conclusion, there are technical difficulties in developing the monoclonal antibody drugs. However, the domestic and international industries have sufficient scientific collaboration to design and produce the accurate monoclonal antibody drugs. The biotechnology advantage and investment in the research continue the development of the novel monoclonal antibody drug in China will soon enter a new era. So monoclonal antibodies can be used for the treatment of the different kind of the diseases. The diseases they are still mocking and challenging the scientific community all over the world, like the cancer, AIDS, and many other viral diseases for which we don't have any other option to cure. We can use the immunotherapy for successful treatment of such mortal diseases. Okay, well, before closing, I would like to salute and greet to my colleague, my friends, and my co-worker, Sherry Bajkar, then Wolfgang, Dustin Hawker, Marwa Al Magharbi, then Robert Heyman, Kunza Noreen, Faisal, Teddy, Ishtiaq, Arslan, and Amin. These are my people, my community. They helped me a lot to make this video today. I also acknowledge the contribution and support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development in Corporation. Thank you very much for watching the video and staying with me for so long time. See you soon in my next video. Take care. Bye for now.